So picture a concrete jungle. I mean, just endless skyscrapers, choked highways, and, a sprawling metropolis humming with the energy of over 23 million people.
That's massive.
Right. This is the greater metropolitan region of São Paulo. But if you look really closely, right at the edge of all that concrete, nestled completely inside the city limits, there's this 186-hectare fragment of the Atlantic forest.
Yeah, it's basically this green island in just a sea of gray.
Exactly. And in late 2017, an invisible threat was lurking right there at the exact boundary where the wildlife meets the city.
And it almost triggered a total citywide collapse.
Which is wild. So to understand how that happened, we're doing a deep dive today into some really fascinating groundwork from a March 2026 paper from Nature Microbiology.
Yeah, we're not going to just, read it like a textbook.
No, definitely not. We're using it as a blueprint to map out the evolution and, the spillover dynamics of a deadly yellow fever outbreak.
Because it really serves as a masterclass in how viruses move across that super volatile border between wildlife and human populations.
Right. And for you listening, understanding this specific forest-urban interface is totally essential.
Absolutely. I mean, we are talking about yellow fever here. This is a disease that carries an estimated 47% case fatality rate in non-vaccinated humans.
47%. That is just staggering.
It's terrifying. So learning how to spot the mechanics of an outbreak before it jumps that border is, one of the most pressing public health puzzles we face today.
And we want to give you those aha moments, right? The actual biological mechanics of how this happens without getting bogged down in all the heavy jargon.
Yeah, let's keep it accessible.
So let's jump right in. We have to start by looking at the very earliest victims of this outbreak, just to understand how the threat was even detected in the first place.
Right, the alarm bells.
Exactly. So the setting is Parque Estadual Alberto Lofgren, and let's just call it Peel to keep things simple.
Sounds good.
So between late 2017 and early 2018, something really tragic happened to the local population of southern brown howler monkeys there.
Yeah, the Alouatta Gariba. So before this outbreak, Peel had this incredibly dense population of these howler monkeys. About 39 individuals per square kilometer.
Which is a lot for that space, right?
Oh, it's huge. To give you some context, that is actually the highest density reported for this species anywhere in the entire Atlantic forest.
Wow.
Yeah, I mean, they were just thriving up there in the canopy. But then, by January 2018, the entire population in that park went locally extinct.
The entire population?
Completely. A near 100% case fatality ratio.
Just wiped out in a matter of months. And, these monkeys, they essentially act as a sort of biological tripwire.
Sentinel species.
Right. Their deaths typically precede human cases by several days, and that gives public health officials a critical warning signal, right?
Exactly. It tells them, hey, we need to start emergency ring vaccination programs for people living right near the park.
They're like the canary in the coal mine, but for a city of 23 million people.
That is the perfect way to describe it.
But, okay, I have to push back on the mechanics of this for a second.
Sure, go for it.
If the howler monkeys are dying out so incredibly fast, doesn't that actually halt the virus in its tracks?
How do you mean?
Well, if the virus is just burning through its available hosts until there are literally none left, shouldn't the outbreak just, starve itself out?
So it does starve out locally eventually, but the sheer density of those monkeys creates this massive window of opportunity before that burnout happens.
Okay, because there are so many of them.
Right. And you have to remember, the virus doesn't jump straight from monkey to monkey. It needs a middleman.
The vector.
Exactly, a vector. So while the monkey population is collapsing, those highly viremic monkeys...
Meaning they are highly infectious, right?
Right, packed with the virus. They're essentially acting as super amplifiers. They're just feeding the virus in massive quantities to the local mosquito population.
Which brings us directly to the vector itself.
Yeah.
Because once the monkeys are gone, the virus is still out there.
Oh, yeah.
It's just flying around on the wings of the primary villain in this story, which is the Haemococcus leucocilius mosquito. And
researchers uncovered something deeply alarming about this specific mosquito population during this outbreak.
What did they find?
Well, Haemococcus mosquitoes are classically canopy dwellers. I mean, their entire life cycle.
So breeding, feeding, all of it.
Yeah, resting, all of it happens high up in the treetops, which, of course, is exactly where the howler monkeys live.
Makes sense.
But during the peak of this outbreak, researchers set traps, and they consistently caught yellow fever-positive Haemococcus mosquitoes down at ground level.
Whoa. So the danger literally took the elevator down from the penthouse to the lobby.
Basically.
And it wasn't just, a few empty elevators arriving at the ground floor. I mean, they were absolutely packed with the virus.
Completely loaded. The researchers looked at the viral load using PCR testing.
Which essentially works like a molecular photocopier,
Yeah.
right? Like you keep photocopying the genetic material until you have enough to actually see it.
That is a great way to visualize it. So when scientists run a PCR test, they look at this metric called the cycle threshold, or sept value. This number basically tells you how many photocopies or, cycles it took before the viral RNA became detectable.
The viral RNA being the virus's genetic instruction manual.
Exactly. So a lower seat value actually means the mosquito was already packed with so much viral RNA that you hardly needed to copy it at all to see it.
Oh, I see. So lower is worse.
Much worse. And these ground-level mosquitoes had a CDEP value that was equivalent to a negative 2.6 difference compared to other mosquito species in the area.
Wow. So they are carrying incredibly heavy payloads of the virus down to the ground.
Yeah, and they also had a minimum infection rate of up to 6.3%, which is extraordinarily high for a wild insect population.
That's terrifying. But what makes this whole situation even more fascinating is why they came down from the canopy in the first place.
Right, the trigger.
Yeah, because it wasn't rainfall, which is what I think we usually assume triggers mosquito swarms, right?
That's a common assumption.
But
yeah.
it was temperature. How does a spike in heat physically change the behavior of a mosquito to the point where it becomes a ground-level threat?
Well, mosquitoes are cold-blooded, so their entire metabolism and their life cycle are completely at the mercy of the ambient temperature.
Okay.
When the temperature rises, several mechanisms basically shift into overdrive. First, their overall development cycle accelerates.
So larvae turn into flying adults way faster. Right?
Exactly. And second, and this is the crucial part, heat drastically shortens what we call the extrinsic incubation period.
Extrinsic incubation period. What is that?
It's the time it takes for the virus to multiply inside the mosquito's gut, cross over the gut barrier, and travel up to the insect's salivary glands.
Oh, so they can actually be transmitted during a bite.
Yes.
So the heat basically turns them into much faster, much more efficient biological syringes.
Yeah, that's a perfect analogy. And because their metabolism is running so hot, they need way more energy, so they start biting more frequently.
Oh, man.
Which drives this multi-bite, multi-host feeding behavior. The mosquitoes start migrating across vertical strata.
So flying from the canopy down to the forest floor and back up again.
Frantically looking for blood meals. And keep in mind, Peel gets 1.6 million human visitors a year.
Oh, right.
Yeah. So a hyperactive, heavily infected canopy mosquito aggressively foraging at ground level. It is the absolute worst-case scenario for human spillover.
It fundamentally changes how you view just taking a casual walk in the park.
It really does.
But if you're listening to this, you might be wondering, well, how do the researchers know all of this with such precision?
Right. The methods.
Yeah, like how do you track viral RNA loads inside a tiny mosquito? Or prove that the exact virus in that insect matches the one that killed a monkey that's decomposing in the jungle?
It's a great question.
To understand that, we have to look at the technology they used, because it actually led to a totally bizarre accidental discovery.
Yeah, the technological shift in this study is vital. Historically, to find a virus, scientists used targeted sequencing.
Like tiled amplicon sequencing.
Exactly. Think of it like having a specific chemical puzzle piece designed to interlock with a known sequence of the yellow fever genome.
Okay, so if the puzzle piece fits, you know the virus is there.
Right, but viruses mutate constantly.
Oh, and when they mutate, the shape changes.
Exactly. And suddenly, your specific puzzle piece no longer fits, and you completely miss the pathogen.
So traditional sequencing is kind of like peering through a keyhole, looking for someone wearing a very specific red jacket.
Yeah.
If they change their jacket, or if they just aren't standing perfectly in front of the keyhole, you just assume the room is totally empty.
That's exactly the problem they face. But in this paper, they used viral metagenomics.
Which is less like looking through a keyhole and more like blowing the door off the hinges and taking a wide-angle photo of the whole room.
Right, because metagenomics is completely pathogen agnostic.
Meaning it doesn't care what virus it's looking at.
Exactly. Instead of looking for one specific virus, researchers just take a sample, break open all the cells, and sequence every single fragment of genetic material present in that biological soup. Then, these incredibly powerful computers sort through millions of genetic fragments to reconstruct whatever genomes happen to be in there.
That's amazing. And that allowed them to get over 99.8% genome coverage of the yellow fever virus, right?
Yes, totally bypassing the mutation problem. They even recovered complete genomes from monkey carcasses that had been decomposing in the humid jungle for 3 days.
That's an incredible technological feat, just given how fast RNA degrades out in nature.
Oh, absolutely.
But because this tool catalogs literally everything in the sample, it resulted in a major plot twist for this deep dive.
The hepatitis A discovery.
Yeah. Inside one of these yellow fever-infected howler monkeys, a specimen labeled NP2754, the metagenomic sweep accidentally reconstructed a near-complete genome of the hepatitis virus.
Which is a profound anomaly. I mean, howler monkeys live their entire lives high in the trees. They do not naturally contract human hepatitis A in the wild.
Right. At first glance, a monkey with a stomach bug just sounds like bad luck. But hepatitis A is a uniquely human problem. So what does finding a human liver and stomach pathogen inside a wild canopy primate actually tell us about the yellow fever outbreak?
It really reviews the hidden infrastructure of the outbreak because the researchers analyzed the genetics of that hepatitis A virus and they found it clustered perfectly with a 2017 human strain.
A strain that was circulating right there in the city of São Paulo.
Exactly. Now, hepatitis A is transmitted via the fecal-oral route.
Okay, so
This means that a wild, canopy-dwelling monkey came into direct contact with human fecal contamination.
wow.
And when researchers mapped where monkey NP2754 was found, it was located just 100 meters away from a park residence.
That is wild.
Yeah.
The human sewage and waste infrastructure literally bled into the protected forest environment.
It did. And this co-infection paints a terrifying picture of the forest-urban edge.
How so?
Well, it proves that the boundary isn't a wall. It's a completely porous membrane. We have urban infrastructure leaking our human pathogens up into the canopy food or water sources.
Right.
And at the exact same time, temperature spikes are driving canopy mosquitoes loaded with lethal wildlife viruses down to the ground.
So it is an extreme, highly active two-way vulnerability.
Exactly.
So, okay, let's recap where we are. We have hyper-efficient ground-level mosquitoes, we have collapsed boundaries where human waste reaches the canopy, and we have a highly susceptible monkey population.
A perfect storm.
Exactly. So to figure out how this cocktail actually exploded, the researchers used all that genetic sequencing to build a phylogenetic tree.
Which is basically a high-tech family tree for the virus.
Right, a map that tracks the virus's mutations to show not just who infected whom, but exactly where and when the virus spread through the park.
And they identified 7 distinct epizootic clusters.
Meaning there were 7 separate times the virus was introduced into the park's monkey population.
Exactly. But if we just look at 2 specific cases, it really tells the entire story of how transmission works here.
Okay, let's look at the very first dead monkey they detected. This was back in October 2017, labeled MP2067. The phylogenetic map showed this was a singleton, meaning it was an infected monkey, but it didn't pass the virus to a single other animal.
Right. The chain of transmission just stopped dead.
Yeah. Why did it stop?
Because that isolated death occurred in a colder microclimate of the park. And it happened during a period where researchers couldn't detect any Haemococcus mosquitoes.
Ah. So without the vector actively flying and biting, the virus was basically trapped in that single host.
Exactly.
So it's kind of like a localized power outage. You know, one house goes dark, but the rest of the neighborhood's grid is totally fine.
That's a great way to put it.
But then we fast forward a few weeks and look at what the paper labels Cluster A.
Yeah, Cluster A.
This viral introduction happened in mid-November. And the environmental context had completely changed by then.
Drastically. Mid-November aligned with a steep rise in temperatures, which, as we discussed earlier, triggered peak vector abundance.
Right, the mosquitoes were everywhere.
The mosquito population was thriving, their incubation periods were drastically shortened, and they were feeding aggressively.
So the environment was totally primed.
Yes. And because of that, Cluster A alone caused 71.9% of all the sequenced cases in the park.
Oh my God.
It literally drove the howler monkey population to local extinction in this incredibly violent transmission chain that lasted only 40 to 51 days.
So if October was a localized power outage, November was a total cascading grid collapse caused by a massive heat wave.
Basically, yeah. It was like a spark landing on gasoline-soaked tinder.
And the math behind that collapse is just staggering. The epidemiological models in the paper estimated the basic reproduction number, what scientists call the R naught.
Right, R0.
They put it at 8.2 for this outbreak. Now, the confidence interval put it between 5.1 and 12.2.
Which is wildly higher than what we see in typical urban mosquito outbreaks for things like dengue.
Wait, really? What does an R0 of 8.2 actually mean in practice?
It means that, in a fully susceptible population, one single infected individual will directly cause, on average, more than 8 secondary infections.
Wow.
Yeah. And when you map that out exponentially over just a few weeks, the case numbers just absolutely skyrocket.
But wait, if you're listening to this and mapping the geography in your head,
Yeah.
we're talking about a park fully encased by a city of 23 million people.
Right.
If the R0 is 8.2, meaning one infection rapidly becomes 8 and 8 becomes 64, how did this not trigger an absolute human apocalypse the moment those mosquitoes reach the park boundary?
It's a valid question.
Yeah.
The key is understanding that the R0 of 8.2 specifically described the sylvatic cycle.
The sylvatic cycle.
Yeah, the loop between the highly susceptible monkeys and the dense swarms of Haemococcus mosquitoes inside the forest.
Oh, I see.
It was an incredibly efficient engine, but it lacked the specific bridge needed to burn through the city.
What bridge?
Well, first off, the rapid extinction of the monkeys removed the virus's primary amplification fuel.
Right, the fuel burned up.
Exactly. Second, to sustain a true urban apocalypse, the virus would need to successfully jump from the forest Haemococcus mosquitoes into the urban Aedes aegypti mosquitoes.
And the Aedes mosquitoes are the ones that actually thrive breeding in concrete environments and urban standing water.
Exactly. And thankfully, no onward urban circulation was detected in the Aedes population.
Wow. We got lucky.
We did. And the third barrier was us.
Oh, the ring vaccination.
Right. Once those sentinel monkeys started dropping, health officials recognized the tripwire had been triggered.
So they immediately launched ring vaccination campaigns for the humans living in those surrounding concrete neighborhoods.
Yes. The alarm went off and the fire doors were slammed shut just in time.
That brings us to a massive systemic problem. I mean, we can't permanently rely on wild animals dying to tell us when we need to start rushing vaccines to city borders.
No, it's far too reactive.
So what does the paper propose we actually do moving forward?
Well, the researchers advocate for a shift toward multi-source surveillance.
Meaning we pull data from a lot of different places.
Exactly. Public health can't rely on a single metric anymore. We must integrate our data. We need to monitor sentinel monkey deaths, yes, but we have to cross-reference that with real-time tracking of rising temperatures and mosquito trapping counts.
Right.
When you have an algorithm showing a sudden temperature spike, a boom in Haemococcus abundance, and an early report of primate illness...
And you know it's coming.
Right. That convergence creates a very brief, highly actionable window where you can intervene before the R0 hits 8.2.
But the paper actually goes further than just monitoring, right? They introduce a conservation concept that, honestly, it sounds like science fiction to me.
Vaccinating the wild monkeys.
Yeah. They argue that because we have endangered primate species, like golden lion tamarins, living in these exact same fragmented, highly volatile forests, we might have to physically intervene and vaccinate the wild monkeys themselves.
Yes, to prevent their extinction and block the spillover source at the same time.
But the math they present for that intervention is incredibly demanding.
Oh, it's brutal. To halt an outbreak with an R0 of 8.2, even assuming you have a vaccine that is 99% effective, you would need to successfully vaccinate about 89% of the wild monkey population to achieve herd immunity.
See, if you're listening to this and thinking, we can barely get 89% of humans to take a vaccine in a brightly lit, sterile clinic, how on earth are we going to vaccinate a jungle? You are absolutely right.
Yeah, it sounds impossible.
We have roads, we have text messages, we have pharmacies. How do you achieve 89% coverage in wild, aggressive primates swinging 60 feet up in a fragmented canopy?
The logistical challenges are monumental. There's no denying that.
I mean, how would you even do it?
Well, you have to locate them in dense foliage, safely dart or capture them without causing traumatic stress, administer the dose, and then release them without fracturing their fragile social structures.
That sounds like a nightmare.
It is. Alternatively, researchers are exploring drone delivery of oral vaccine baits. But even then, ensuring equitable consumption among the troop is really difficult.
Because one monkey might just eat all the bait.
Exactly. However, the authors argue we are rapidly developing the toolkit to make this possible.
Like what?
Well, we now utilize acoustic tracking. We are literally placing microphones in the canopy that use AI to analyze specific howler calls.
Oh, wow.
Yeah, so it maps monkey densities and movements in real time. We also have low-cost, rapid field diagnostics now.
Right.
And we have robust citizen science reporting apps where locals can log dead animals with GPS coordinates instantly from their phones.
So it is this massive, high-tech race against rising temperatures.
It really is.
Man, we've covered a lot of ground today, so let's pull all these threads together.
Sure.
We started with the tragic, rapid extinction of howler monkeys, acting as the ultimate biological tripwire for a sprawling metropolis.
Right.
Then we learned that the threat isn't safely confined to the treetops because heat drastically accelerates mosquito metabolism, turning these canopy-dwelling insects into hyper-efficient, ground-level vectors loaded with the virus.
The elevators down to the lobby?
Exactly. We saw how metagenomics didn't just sequence yellow fever, but cataloged everything, revealing a wild monkey infected with human hepatitis A.
Proving that urban sewage and wild ecosystems are bleeding into one another.
Right. And finally, we unpacked how a localized viral introduction turned into a devastating transmission chain with an 8.2 reproduction rate.
And it was only stopped because the biological fuel burned out and human fire doors were shut.
Right. It fundamentally proves that the forest-urban edge is an incredibly active, volatile landscape.
It truly is. And honestly, it leaves us with a necessary shift in how we think about these outbreaks.
What do you mean?
Well, in epidemiology, we constantly rely on the word spillover to describe a pathogen leaving the forest and invading our human infrastructure.
Right, the virus spilling over into us.
Exactly. But when we look at monkey NP2754, living up in the canopy but infected with a human liver virus originating from our own waste, we really have to reconsider that narrative.
Because we spilled over into them.
Exactly. We must ask, are the viruses invading our cities? Or has our urban sprawl finally pushed so deeply into the forest that we are now active participants in the enzootic cycle?
The wild animal-to-animal transmission loop.
Exactly. We are a part of it now.
So when the boundaries collapse completely and pathogens are freely riding elevators up and down the canopy, who is really invading whom?
It's a great question.
That is the exact question we need to be asking. Next time you look at a map and see that sharp, jagged line where the concrete jungle stops and the green forest begins, remember, the reality on the ground isn't a clean border.
Not at all.
It's a deeply connected, highly reactive landscape, and the invisible threats are moving rapidly in both directions.